Hey, good morning, everybody.
Good morning. Good morning, Tom. How are you doing? Good morning.
Doing very well. Hope y'all had a great weekend.
Yeah, I had a really great weekend. How about you?
Yeah, I sure did.
Perfect.
Yes, I did.
Awesome. Oh, hi, Rachel. How are you doing?
I'm good, thank you. I'm glad to be here.
Sure. Hi, Ali. I see Ali here. I see Terry. Good morning, Terry.
Good morning, Terry.
Good morning, everybody.
Good morning. Hello, Adam. So let me make all of you a co-host. Okay.
I wonder what number we're going to climb to here with all the people who've signed up for this.
I guess we have accepted something close to 107 participants, but I don't know how many of them will join in the morning.
Yeah.
So Tom, I prepared a little intro, so let me know when you want to hear it.
Okay.
In fact, I'm also going to be using breakout rooms.
Oh, you are?
Am I empowered to create a breakout room?
Actually, we made 6 breakout rooms, which Caleb, which I introduce him in 1 second, he will let all of you inside any of these 6 rooms that you want. So Caleb, are you available? Oh yeah. Okay. So
Not now. I'm just saying to make sure that to alert you to set one up, but a little bit later. I'll let you know when.
Sure. Perfect.
And I think we need, I think no more than 5 per breakout room. So, compute that when you, once we get to that point.
Okay.
Oh, we just lost somebody. Somebody just went down from 32 to 31.
Yeah, sometimes people have trouble connecting and they have to try again, maybe.
Right.
Yeah.
So I just wanted to say hi to everybody and say I'll see you in 2 days.
Okay.
Oh, hi. Yes. Hi, Bob.
Hi, Bob.
Ted is also on, and he will be. We will be running the session starting on Wednesday. So
Perfect.
that was all I wanted to say. Let you guys go about your business.
Okay.
I look forward to working with you guys in a couple of days.
Sure.
All right. So,
Us too, Bob. Thanks.
Yeah, so this is a tag team here this week, and this is just the first third. So I'm going to launch with share screen, if that's okay with everybody.
Sure.
Okay, here we go. This the breakout room seems to be taking up the bandwidth here. Yeah. Oh, I can get rid of it, I guess. Okay. So welcome everyone. This is an old slide of mine that goes way back. You can tell because if you look at the picture of Tom Bartol here, you can tell that he's a lot younger. And I put this together from a very early simulation of the release of glutamate. And as you can see, in this era, we had a block diagram for synapses pre and post. And the little yellow things here is the glutamate. But this is a historical interest because the origin of MCell goes back to Cornell and Mika Salpeter, who was Tom's mentor for his PhD. And the math for it came from her husband, was a distinguished astrophysicist, Ed Salpeter. And so Tom came to my lab and we continued it in conjunction with Joel Stiles, as you can see down here, who moved to Pittsburgh Supercomputer Center. And so, you know, so this is the team. This is Team M-Cell at the very beginning, probably, Tom, was probably 30 years ago now.
So I believe this was like 32 years ago.
Wow, okay.
Yeah.
Okay, well, this has a lot of history. And in fact, I think, you know, you'll see that M-Cell is now up to version 4. Version 0 was in Fortran, right, Tom? This was the original.
Yeah. So in this, when this picture was taken in 1989, we were at version 0, or it wasn't even called M-Cell at the time. We didn't pick the name M-Cell until 1996, when we were trying to get grant funding for it. We had to come up with a good, slick name. But in 1989, it was called MEPC Sim, for Miniature End Plate Current Simulator. Because what we were simulating at the time in Mika's lab was the end plate, the neuromuscular junction of lizards. And I did my thesis project using 2-electrode voltage clamp to study the synaptic current generated in the neuromuscular junction. And that synaptic current was called a miniature end plate current.
Okay, so Tom, you know, I don't want to give all the history here. I just want
Right. Oh, okay.
to indicate some of the collaborators we've had over the years. Kristen Harris in particular, I want to give her a shout out because she provided us with the next generation. And instead of this block world of the brain, we got a reconstruction, an EM reconstruction from her that really you know, put us into the 3D business for realistic geometry. And the other person I need to shout out is Dan Keller, who was a bioengineering graduate student at the time. And I want to just show you one slide from that era. If I can, let's see, go on to the next slide. Okay, what you're seeing is something that is impossible to see without a computational reconstruction of the neuropil, of a very detailed neuropil, of a very tiny piece, 6 by 6 by 5 cubic microns of the CA1 neuropil. And what you're seeing here is not what you normally see, which is, you know, the dendrites and the axon, the synapse. What you're seeing is the extracellular space that Justin Kinney reconstructed. And I won't go into the detail, but it wasn't, you just can't take the raw data because during the embedding in plastic and alcohol dehydration, the extracellular space shrinks, so you have to expand it. And it really just does, it has a lot of information in it. And let me just point out what the colors mean. The color means, if it's blue, it means that it's a surface between 2 neurons or 2 neurites. And this is, it's like a sheet. In other words, it has the topology of a sheet. Now, what about the yellow circles here? Well, those are the locations when 3 or more neurites get together, processes, and you know, axons and dendrites and glia. And when they do that, then you get something that looks more like a tunnel. So what you can see is that the extracellular matrix consists of tunnels, connected tunnels, that actually are large enough to shuttle very large molecules around the neuropil. And before this, there wasn't any good explanation for how that was done. You know, we're talking about very, very large proteins and things. So this really, I think, is a, for me, it was very revealing. Okay, now I'm going to show you a movie. And one of the nice things that came out of developing the, you know, the visualization was that we could make movies of the simulations. And I don't think Tom is going to show you this, so I decided I would. And this, again, is from that reconstruction, and this is from Justin Kinney. And we asked the question of what would it look like if you were in the CA1 region when you were asleep, when in this case, a rat was asleep, and the CA3 had just unleashed a sharp wave ripple, which is a burst of action potentials lasting about 100 milliseconds. And it's going to bombard this tissue. And what you see in the background are the ghostly, you know, synapses that are going to be releasing glutamate. Okay, so here it goes. And we've orchestrated it so that I hope that this will come through. Oop, it didn't seem to want to play, but let's try it again. Here we go. Okay, that white is the balls are glutamate, and the yellow is actually when the glutamate binds to an astrocyte. That's how it's uptake into the astrocyte. So that was one release. Now we're going to back up and see what the rest of the nerve pill looks like. The red dots are the dying embers of the glutamate being taken up by the astrocyte. Okay, well that was the fireworks, which definitely more dramatic with the music. Hope you appreciate the orchestration. There was very closely timed. Although it's completely random, by the way. I think we just picked a piece of music and just pasted it right on. Okay, so now here's the breakout room. Here's what I want you to do now for the breakout room, and I'm going to stop sharing. I'm going to tell you a story first, and this is a true story about Erwin Neher, who all of you probably know was one of the inventors of the patch clamp, right? I mean, incredibly important for being able to record from single ion channels. So Erwin Neher was invited by the Pope to a Pontifical Academy conference in Rome, and they were all, you know, all these very Nobel Prize winners and distinguished scientists were all asked to come up with a question that they would ask God, you know, important scientific question. And here's the question that Erwin Neher came up with. What are all the calcium binding proteins? And what are their reaction rates? And we still don't know. In fact, we know we have identified a few of them, but the fact is that there's not a lot of work that's been done on these calcium binding proteins, but they're incredibly important for regulating, obviously, the entry of calcium and the activation of various mechanisms that, like the release of neurotransmitter. Okay, what I'm going to ask everybody here, okay, is when you get to your breakout group, I want you to agree on a question that you would like to ask God, right? If you had one question to ask God, what would it be? You've got to all agree on the same question, okay? And you have 10 minutes to come up with a question. And then when we come back, we'll all share those questions. And I would, you know, I'm really curious to know if there's any, if there's a question that that more than one group comes up with, because then that really tells you that it's an important question. Okay, so Rosita, could you break us into, I guess we need probably, let's see.
We have 43.
Yeah, I would get 9 rooms.
9 rooms. Okay, so let me ask Caleb and see if he can make new rooms. Caleb, would you?
Oh, no. If
able
it's too much work, just go with the 6 rooms. That's okay.
I got it all figured out. I think all I have to do is hit open and everything should work.
I mean, we probably need 9 rooms. So would you be able to add 3 more rooms, or I should do that?
I have it.
Excellent. Thanks, Caleb.
Excellent.
Got it.
Okay, there we go. Do I have to do anything to get the?
No, you just find a room
Okay.
that you want to go in and just join it.
No. It should be random assignments. It's got to be random. Just
Okay.
random assignments, and then get 10 minutes. After 10 minutes, we got to come back. So we're going to depend on you to guide us through this.
Cool. Okay. And
Give me
let
1
me
second.
again ask Caleb, would you be able to randomly put people in those 9 rooms? Thank you.
Yeah.
Now remember, this has got to be the most important scientific question now
Right.
that you can ask. Within the bounds of MCell that we're going to be talking about.
Here we go. Sorry, it took me a second. Got it figured out. Looks like Tom got into one of the rooms.
Hi.
Hi. Hello, Susanna. You just joined?
Oh, looks like she got into one of the rooms.
Oh, okay. So, Caleb, where I cannot find my room.
So yeah, I think because I created them, they're different. I don't know. So I have all the rooms on my screen.
No, I mean, where should I join? Or
Oh,
shouldn't I?
just you can join wherever. I can't find you to
Okay.
assign unless you're already somewhere.
Okay. I can jump in one room. Do you want to join and ask your question? I can stay here and whatever you prefer. Do you want to join?
No. I'll probably just stay right here. I'm interested to see what everyone comes up with though.
Cool. Then let me join one of these rooms.
Okay, we need to stop the breakout room so that we can all get back.
All right, I'll do that right now.
Hey, Jim, how are you?
Good.
Good.
You-
We just had the break. Let me give you the background. So I asked everybody in the breakout room to come up with a question that they
I
would.
was in it. I was here.
Oh, you were there. Okay, good. So
I was expecting like cat pictures, and then I came on and you're asking God questions. I'm like, okay, damn, I have to like turn on my brain already.
right. Okay. So I'm going to have to actually have to introduce a speaker at another meeting, unfortunately, at 8. It's one of those things that only Zoom can make possible, is actually to give 2 talks back to back, right? 2 different meetings.
So, yeah,
Rosita, do you know what just happened?
People just,
The
you just
breakout
sent us back to the breakout room.
reopened.
oh, you put us back to the room? Yeah.
That my mouse was on my other screen. I must have misclicked something.
Oh,
That my bad. That was totally on me. I swear my mouse was on my other screen. I don't know how I did that. My
Okay.
apologies.
Okay.
I'm
So-
closing this window.
oh, okay.
Okay, well, I think we're not all back yet. Is Tom here? Good. Okay, Tom's here. Okay, so
Is this
Tom,
the
I
right
have
room?
to go away at 8 to
Okay.
introduce a speaker at another meeting.
Okay.
So I'm gonna pass the baton to you, but I just have to say we had a great discussion, and from all over the world. I mean, it's amazing. India, Jordan, Netherlands, Germany. I mean, it's really quite a network. And that one thing I noticed was that everybody thought that the most important question was the problem that they were working on.
Oh.
Surprise, surprise. So we don't have a consensus, but I'll tell you the question I had was, and actually someone else had it, I forgot, I think maybe it was Moritz, which is what's really going on in the PSD? And is there really a liquid phase transition going on there, right?
That's a good question.
Yeah. Okay, well, I'm going to say goodbye to everybody and pass the baton to Tom. And so
Here, I have a question for you though, Terry.
okay.
And so this is not as, I do have more specific questions, but the one that I came up with was, is there another way to make life besides, central dogma as it exists on Earth?
Yeah. And that's related to the question our group came up with, which is,
Okay.
what is life? What is life? But I'll let.
Okay. Well, actually, I have to say, I restricted the question to the synapse in our group, but why not? Why not? Francis Crick once asked God, why do flies have imaginal discs? It's an organizing center during development. And so God told him, why Francis, no one has ever asked me that question before. I've been putting them into flies for 200 million years. I haven't had a single complaint. We took a little bit of this from here, a little bit of that from there, and put them together. Actually, I don't really know, but it seems to work. Okay, well, goodbye, everybody. Take care.
Bye, Terry. Thanks for coming, Terry.
Thank
Bye.
you very much.
Bye-bye.
Bye-bye.
Okay, so our breakout icebreaker this morning was a bit of a departure from what we did last year, but it was because Terry could only join us during the icebreaker this morning, and he thought it was important to kind of get us kicked off the right way. So thanks for coming and joining and participating in that wonderful little diversion there. And I think it was a great way to get us in small groups to meet each other and have these great discussions. So I think maybe what we could do quickly is maybe just say what we thought what our agreed upon ultimate questions were. And so since I was in a breakout room with Wouter and Cheval and Grace and Susanna, we designated Grace to be the person to discuss our question. So Grace, give a brief synopsis of our question.
Yeah, so basically our question is kind of a fundamental question, which is we would like to know God's definition of living thing. Because since my team is working on bacteriophage, so we know that bacteriophage is one of the viruses. So I'm kind of curious of how God defined whether viruses are living things or non-living things, and yeah, we're kind of curious how life began from the universe, and so yeah, that's basically our fundamental question. Yeah.
Okay, so I'll let the rest of you organize yourself, and someone should step up and from your group and talk about your question. So that was group number 9. I think we were in group breakout room 9. So maybe we can go in order. Who's someone from breakout room 1 should, if you know your number. I don't know if you took note of your number, but if we go in order, breakout room 1, please, someone.
Yeah, I can speak for breakout room 1.
Okay,
So the
thanks.
question we had was like, we would like to ask God for a mathematical formula which will take in a protein sequence and output a protein structure, which we hope will be a foolproof and does not really rely on any heuristics. And then everyone can happily live ever after.
And I think there's a new machine learning algorithm that
Yeah.
almost does what we're talking about. Cool. Okay, breakout room 2.
Oh yeah, maybe I can say it again. Yeah, we were the synapse guys, and we had the question like, how does this organization of the synapse of the PSD come about with all this patterning, segregation of different redundant molecules in there? How does it come about? How is it maintained? And yeah, what are the principal like mechanisms like liquid-liquid phase separation, etc.?
Yeah. and with something that self-organizes like that, is it alive? In the way that a virus is alive? I don't know. Okay, room, what, 3, I guess, 3 or 4.
Hey, can you hear me?
Yes.
So we talked a bunch about consciousness, and I think we figured to ask that what's like the minimum requirement for attaining consciousness, and what's the maximum scale that consciousness can be achieved? Like, is it, can galaxies gain consciousness, and can individual little cells have consciousness, stuff like that? I don't know how it relates to M cell, but maybe it can be modeled.
Well, is there any limit to what can be modeled? I mean, maybe theoretically not, I don't know. I guess it, so, you know, Turing has, and in mathematics, there's this definition of computability. And is consciousness computable? I don't know. There are things that are not computable. It's hard to imagine because the tools of mathematics break down when something's not computable. But we know that it's been proven that there are things that are not computable. There's a mathematical proof that
And
math
Gödel's
is,
theory of incompleteness probably suggests that it's not computable.
yeah, I think
Right,
so.
because if you can modify yourself, you shouldn't be able to just start from axioms and compute the end result.
Yeah. Roger Penrose has a whole, a whole thesis, thought thesis on this problem, and he's a firm proponent that consciousness is not computable. Okay, next breakout room.
We on 4 yet?
Yeah.
I think ours was 4. I don't want to speak for the group, but I guess I will. So we initially, our initial question was, what is the minimum level of abstraction we would need in order to be able to understand brain functions such as memory? Perhaps you could make that kind of the other extension, the other arm of memory consciousness. Then another group member brought up a more fundamental question: is what is, but somewhat related, is what is are all and related to the last question, are all problems solvable in polynomial time? And so they're kind of related questions in that if the maximum level of abstraction is too high, the answer may be, and if not all problems are solvable in polynomial time, then maybe we'll never solve things. But I guess an easier question would be, what should I study in biology using MCell? So that might be a question I would ask. So.
Cool. Okay. Next ultimate question. I think we're at 6 now, I think, right?
We were 4. So I think
you
we're
were
5.
Okay, so let's hear from group 5. If you know your group number.
Yeah, I was in group 5 with Ali, Mariam. Oh, I forgot the 3 other people's name. Sorry. So someone from our group, go ahead and ask one question. We didn't, decided about the which question we ask. I mean, who want to go and ask?
I'll with one. We didn't get to a consensus. We didn't have time.
Yeah.
But what I would ask is what's the minimum amount of interactions that you need to be able to get something like the genetic code? So
yeah.
Minimum amount of moving pieces.
Yeah, that's like an origin of life kind of question. And I've thought about that myself a whole bunch. And I'm sure all of us here have given quite a bit of thought to this, because it's really one of the root questions in biology, I think. Okay, next up.
Room 6.
Yeah. Let's hear from group 6, if you know your number.
I think it was us, and we had a great discussion with Hania, Sean, and Don, and it seems like pretty much most of our team members were physicists or physicist background.
Okay.
And I'm a biologist, and I believe Hania is now working on something that is also deals with biology, but she's also a physicist. So we kind of like had a mosaic of pulling everything different directions, but ultimately come up with a question: how to define or how to identify, how to differentiate between a living and non-living system, or bring it kind of a go back and forth in a sense. That's what my feeling about, because we got cut off with all this, going back to the main room, so we couldn't finish our thoughts, but we kind of, that's what I hope I'm getting out correctly. And team members, please correct me if I'm wrong. Sorry for stepping up and speaking out on behalf of the team. I think yeah.
I think that's right.
Yeah, thanks, Don. Thanks for correcting.
Okay, thank you, Dima and Don. Okay, group 7.
I think that was me and Josiah, and we had a few other people, but Josiah and I were the only ones who talked. And we threw around, I think we hit some of this similar territory to what other people have brought up. I said our question to Terry was, are there other ways to encode life, which also suggests this question of what's the minimal encoding that you need, or what's the minimum set of tools you need, mechanisms you need to encode life. And so another interesting question that I think is sort of related to what we're trying to do here is sort of, what are the minimal abstractions for understanding what happens in a cell? And that question has many potential layers to it, because in some sense we know the chemistry, but we don't really quite, and we know that, we know how things get built up. We know structures like the postsynaptic density even, but we don't really know what they're for in all cases, or how they're sort of at a higher level, how they're functioning. So those are all questions that we can think about a little bit as we build networks and spatial models and look at images of cells and so forth.
Yeah, in our group, we talked about this thing of networks. It's networks all the way down and networks all the way up.
I forgot to turn on the camera.
Okay, group 8.
I believe we were group 8, and by we, I mean Bernard, Stephanie, Juhong, and Michael, and myself. Love all of your questions. We were more generic than all of you were. And so I think we concluded that our question would be, what are we missing in finding a cure for cancer? As broad as cancer is, there has to be something that we are missing. And I guess it goes back, if we want to talk about the biology of this, it goes back to how do you regulate the central dogma, basically, right?
Yeah.
I'm studying non-coding RNAs, so I'm sure there's something there, but like we said, everyone cares about what they're studying, so that applies to that.
And it's funny how biologists, researchers come up with names for things like non-coding RNA. Like, what does that mean? It codes for something, and somehow it got made.
Right. It doesn't code for protein. That's the main thing.
Right. Yeah.
Biology is very protein centric at the time.
Right. Yeah. Exactly. I guess the central dogma wouldn't be dogma if it weren't dogma, right?
Right. And Michael posted on the chat the informational integrity as well. That sort of kind of also applies to the cancer, I guess.
Yeah. Okay, group 9.
Didn't you say you were group 9?
Oh, thank you. And another question for God is, why do we, get airheads sometimes? Anyway.
What group?
Okay. What's that?
I said, what group am I in?
Yeah. What group am I in? Or comfort. Yeah, exactly. Okay, so I think this was a great little exercise that Terry had us perform, and I think we have really successfully achieved what he wanted us to achieve. So, which is, you know, to break the ice and get to know each other and start a conversation around everything related to life and living things and networks, all the way down and all the way up. And a fundamental tool that our group has, our team has developed is ways of looking at networks of interacting molecules. So with that, I'll hand things off to Jim, who's gonna tell us more about that.
Okay, wow. Yeah, I'm happy about that icebreaker because it forced me to engage parts of my brain that I feel like haven't been engaged in a while. But also, it was great because there's a lot of people who are clearly thinking about diverse things and thinking deeply about many different things here. So I don't know where everyone is from or what they're doing exactly, but I know that they're thinking. So I guess that's probably the most important thing. We do have a fantastic group here, and I guess we're up to like 61 people total on the Zoom now. And I do say that thinking about these deep questions, we're going to be pondering much less deep things for a lot of today. So I really hope we can be useful and that you guys can learn something new today. And it's always a little bit intimidating to try to figure out what kind of an audience we're going to have and what would be useful for people to learn. But I guess I'll start with just a very brief overview of who we are who put on this workshop. And let me, this is the BioNetGen intro. So here is the presentation, and we'll always have to get Zoom's complexity always confounds me. So let's see if I can get this to work. This top one, is it going to share the right thing?
Yes, it works perfect.
You're seeing my screen.
Yes.
Which says welcome. And so I can officially say welcome at this point to the 9th annual Bio Cell Modeling Workshop. And your instructors, you have met Tom already, Tom Bartol at the Salk Institute. And myself, I'm James, or Jim, I go by Jim most of the time, Fader at the University of Pittsburgh, and Bob Murphy, who'll probably be joining us on Wednesday at Carnegie Mellon, are the 3 main instructors for this workshop, sort of head instructors. We will be talking about the various tools that we develop under the auspices of this National Center for Multiscale Modeling of Biological Systems, MMBIOS, as it's called. And we'll be doing that for the next few days, introducing you to the tools for the first 3 days of the workshop. And then the last couple of days, we'll be doing some more advanced topics and working with groups potentially developing their own models as well. I want to say first off the top, a big thanks to Rosita at the Pittsburgh Supercomputing Center. She has been the central person who's been organizing this workshop, making sure all the logistics are running smoothly, getting all the information out. And so big thanks to her, and she'll be, behind the scenes for a lot of this time. So we really appreciate the support that she and the Pittsburgh Supercomputing Center provide for the center as a whole. And that includes both the logistical and computing support, but also scientific engagement as well. So I don't want to suggest that it's just logistics going on. Okay, and all of us are assisted to get in the right window by a lot, varying teams of varying sizes as well, who are going to be either working manning the Zoom and various breakout rooms to help people as they encounter technical questions along the way. So I have a number of people from my lab. Sinan is the chief developer of the software tools that we'll be talking about through most of today. And so he'll be able to help people out. He's also going to give a presentation later on Jupyter notebooks and working with our BioNetGen tools in that context, and all the exciting workflows that you can develop there. And then Tom has a big team that have just put out a new release of MCell, which is like brand new and shiny for this workshop, which means you guys are getting like a first gloss, first chance to jump into this really exciting new platform that's been really rebuilt from the ground up. And Adam Hussar is the lead developer of that effort and has done just amazing work to put that together. So really excited to get to see all of that stuff, primarily starting tomorrow. And then
and try not to break it too badly.
Yeah, this is going to be this. We should call this like this is like in a way, this workshop is kind of a throwback to some of our earlier days when we had less mature software tools. We had some very mature tools that we've been using for the last 4 or 5 years, and we decided to throw all caution to the wind and build up a whole set of new ones that we think are pretty well tested, but they're not battle tested. They're not workshop tested. So this is going to be an interesting experience for all of us. So with that said, do bear with us if there are technical difficulties along the way. That could be the price.
Susanna's giving us in the chat. Susanna's giving us her evil laugh.
Oh, I have lots of stories of terrible things that have happened for live demo. Oh, why that happened. Live demos. So I'm well aware of all the risks. But
in the very first MCell workshop that was held in 1996, Joel and I thought we knew what we were going to do and had planned ahead. But after the first day and the questions that the students were asking, we realized we needed to revamp everything. And so we did like 4 all-nighters in a row, because it was a 5-day workshop. So we ended up doing like 4 all-nighters in a row, Joel and I together, redoing everything and even fixing the software and all kinds of stuff. It can be like that sometimes.
Yeah. So so there's excitement ahead, but I think that hopefully it'll all be worth it, and we'll be able to convince you of that. Okay, so and then Bob has Ted. Ted will be, I think, doing some of the hands-on work with the cell organizer tool as well on Wednesday. And I put ... because there's a large number of people who have contributed to the effort behind cell organizer, and I just didn't have their names handy to put on this slide this morning. But all of these tools are the efforts over no matter how much I work with Zoom, I still struggle with certain basic functional aspects. Changing the slides, Zoom and PowerPoint, I don't know, they need to play better together. Okay, so let me tell you a little bit about this, about the center, the Multiscale Modeling of Biological Systems, or MMBIOS Center. Essentially, this is an NIH-funded center that is headed at the University of Pittsburgh, but involves researchers around the country at both coasts, basically. And the overarching biological themes are spatial organization and temporal evolution of signaling, both in the central nervous system and in the immune system and beyond. Yeah, we have Pitt, we have Carnegie Mellon, Salk Institute, and Pittsburgh Supercomputing Center. Yeah, and we've been collaborating together now, working together now in this form for, this is our 9th year, but this collaboration actually goes back the decade prior to that, and really started as a collaboration that, between the Pittsburgh Supercomputing Center and Salk, focused around the development of MCell. So I'm going to skip this generic background, I think you guys are all familiar with the ideas of using modeling and simulation to connect big data and knowledge. And I just want to focus a little bit on the software. Oh, okay. And connecting scales. We had a really nice introduction to that from Terry and the movies that he showed. The protein dynamic scale is the sort of the one scale we won't quite reach in this workshop. We're not going to talk about modeling individual proteins here. There was actually another workshop that happened week before last, I think, on biophysical modeling, and that involves yeah, okay. I always forget how much animation there is on each of these slides. Okay, so these are the four main sort of central tools and sort of that encompass different scales that are developed under the center. And as I said, we'll be talking about three of them today. The molecular modeling, as I mentioned, was the subject of the biophysical modeling workshop that was held a bit earlier. All of our tools we're working towards integration of that scale as well into the kinds of modeling that we do, and we have a number of research applications already in that direction. But as far as the modeling workflows that we're going to talk about this week, we're going to talk about spatial modeling, spatial cell modeling using MCell. We're going to talk about reaction network modeling using BioNetGen. And we're going to talk about deriving molecular geometries and protein distributions from images and developing models that can be then incorporated, where we can connect all of these tools, MCell, Cell Organizer, and BioNetGen. And so this diagram shows one possible way to connect these various tools in which we develop biochemical network models using BioNetGen. We can develop spatially resolved simulation models using MCell, and we use Cell Organizer to derive essentially what are called generative models of cells and cell geometries in order to provide the actual configurations, the spatial configurations for MCell, the locations of proteins that are involved in the network dynamics. And we can couple all of these together and make models that can be simulated in MCell that encompass really a huge range of complexity. And so we can use models of that sort to investigate the effects of for example, geometry and spatial distribution on cellular dynamics. And then the fact that we're developing a whole suite of different tools that can be made interoperable in various ways lets us combine these tools to create new algorithmic approaches to problems. So here is one tool that we've developed over a course of years that enables us to investigate the effect of cell geometry and on reaction network dynamics using so-called rare event sampling, in which the amount of simulation time that's required, potentially required to observe complex phenomenon may be beyond the range of what's feasible with a single simulation trajectory, or even an ensemble of trajectories. But we can use enhanced sampling methods to focus on the dynamics that are of interest to us. So because we're a center that develops multiple kinds of tools for modeling and simulation, we can combine advanced capabilities in each of these areas to develop new algorithms. And the rare event sampling methods that we've been developing, we actually have, if you look on the BioNetGen website, which we'll go into in a little bit more detail later on, you'll see examples of where we're developing capabilities based on our Jupyter notebooks to actually set up in a very simple way these kinds of rare event sampling simulations and look for example, transitions in genetic networks, transcriptional networks. Okay, so there's lots of things that are going on with BIOS. We have a nice website. that has listings of a lot of different activities that we're involved in. So check us out on mmbios.org, and then all of the software tools have links through that website as well. So if you get lost, if you forget how to get to one of our tools, the mmBIOS is always a good staging area. Okay. Okay. And I think that does it for the basic overview presentation. I want to take just a 2-minute break for me to grab some water, because I know that I'm going to, my voice is going to run out fairly quickly if I'm not able to do that. So give me just a minute and I will be right back, and we can jump into some BioNetGen.
Maybe while we wait for Jim, I wonder if someone might have a question a burning question they have.
I guess I could ask Tom, has it been hard to update Cell Blender to work with Blender 2.93?
Not too bad. Was less difficult than I thought, and it popped into my head to try to do it about 3 weeks ago. So
wow.
So of course, Adam has been working very, very hard on MCell4, and it works in the old Blender 2.79, and we've been using that for like 1.5 years in the lab as we continue to develop it and debug it. But then I thought, well, okay, it's all together, it's all working, and it's all brand new and shiny, but we really should be using the latest Blender for this workshop. So I thought, well, we have a backup plan. We could use the old Blender, but I said, I might be able to do it. And so I started working on it about 3 weeks ago, and it actually wasn't as hard as I thought. So after about a week, I said, I think we can pull this off. So, and I think it's pretty much there. So we're going to find out.
Oh, that's amazing, actually.
Thanks. Okay, we'll let Jim take us into the world of BioNetGen.
Okay. That worked, right? Okay. Let me just check one thing here. Okay. Well, hopefully we'll be able to, when we get to the hands-on portion, be able to switch back and forth between my slides and VS Code. That's the main thing. And we'll see. It'll be very interesting to see. Actually, one of the big catch points of doing the workshop on Zoom is to figure the interaction between all of the different interfaces that we're trying to present and zoom. So I hope this Cell Blender Zoom interface works well. It's not, these things are not always straightforward. Here, I'm gonna.
Yeah.
I have some coffee left too. Okay, so we're gonna start out really basic, and I hope that this material is interesting to at least and useful to a subset of the participants. I'm sure for many of you, you guys work with reaction networks all the time. And so some of this is going to be review and kind of basic, but I want to make sure everybody is on the same playing field here and has an idea about what's going on and what's underneath the hood in terms of the basic modeling methods that we're working with. And then we're going to jump into sort of nuts and bolts stuff, how to actually set up models using this, as I mentioned, this new interface that we developed for BioNetGen that's based on VS Code. And I spent an inordinate amount of time making the slides, getting into a workflow of making the slides to demonstrate all this stuff. So I hope it all turns out to be useful to you. Okay, so this basic abstraction that we're going to be working with for understanding cellular dynamics on the first day is the reaction network. And reaction network models can be used to describe all kinds of different processes from atmospheric chemistry to epidemiology to molecular biology. And so we'll actually be do our simple demonstration examples of reaction network modeling in this epidemiology level. The inspiration for that, I think, is pretty obvious. We'll talk a little bit about SIR models, not into a whole lot of gory detail. Most of what people, I think, are probably interested in, at least who want to model the origins of consciousness and the postsynaptic density, are going to be interested in the molecular biochemistry level. Finding the pointer is always the hardest part about this. I'm trying to get right click. Oh, I know it. We'll have to see how this goes as the day goes along. I sometimes get very weird lag things happening between Zoom and PowerPoint. So hopefully that doesn't get too crazy. Okay, so signaling networks. So we'll talk a little bit about how to encode signaling networks as reaction network models. As we dig into things, and really sort of my interest, the place that I come from in terms of my scientific background is at this molecular scale and trying to describe the interactions that are taking place in the context of cell signaling, at a more refined molecular level of description. And we'll see in the second presentation that I give about that digs into rule-based modeling, how we can use this BioNetGen framework formalism to make more detailed models of biochemical processes, which I think is one of the main things that we've been involved in partnering with MCell to make it possible to make detailed biochemical models in a more scalable way within MCell models. So we've worked now for nearly a decade on bringing BioNetGen and MCell together, and hopefully we'll get to see some of how that works through this workshop. Okay, so the reaction kinetics start at the level of individual reactions that come, the notation comes from basic chemistry, but it generalizes into many different contexts. So here in our SIR model, where we have susceptible individuals, infected individuals, and eventually recovered individuals, we have the basic infection event is described as using so-called law of mass action. So the basic terminology, S and I, are species. So they're entities that represent pools of identical or indistinguishable entities that can come together through some generic interaction and change their nature and convert to other types of species. So in this reaction, a susceptible individual becomes infected through interaction with an infected individual. So you start with one susceptible and one infected, and you wind up with two infected. And this parameter beta is the rate constant that describes the rate of that reaction. Here, we're going to think about rates as constant entities. They're given by a number, which may have units associated with it. As we get along later on, we can think about generalizing the concept of a rate constant to a rate function, which may be a function of other parts of the system as well. But for now, it's just a constant entity. And so the rate of this reaction is given by the product beta times the number of susceptible individuals times the number of infected individuals. And this is the simplest description that we can give for this kind of a process involving mass action kinetics. And the assumption is as described there. At some point, I'm going to have to switch the resolution on my monitor to hopefully get rid of the weird latency that I'm getting here, but we'll keep going as long as it's feasible. Okay, so reaction network models are built from individual reactions. So we have this, we're going to really talk about the abstraction that's used to build up complex models. Here we have a second reaction. So first we had the infection, the reaction that describes infection, and then the second reaction is the important one, where the infected individual becomes recovered. And overall, we have a total of 3 species in this network. I should mention that this second reaction is also governed by mass action kinetics. And what we do when we make a reaction network model, we want to track the time evolution of species. So how did the number of susceptible, infected, and recovered individuals vary in time? And the complete model consists of reaction species and rate constants, as well as the initial values for each of these entities. So I'm just formalizing that here. And then one way we can describe the dynamics of the system, and not the only means possible. And what I'll say is I noticed that there's a question in the chat. Yes, the PowerPoint slides will be shared, and actually they are already available. Maybe someone could post a link to the right. The slides are actually in the subdirectory BioNetGen. Yeah, there we go. Beautiful. Right. So the slides that I'm actually presenting now are available there. And if there are updates, I'll post updated versions there as well. Okay, so the ordinary differential equations are one way to describe the dynamics, and they will describe the mean behavior of a network. So assuming that the concentrations or the numbers are continuously distributed in time, and that the ordinary differential equations will describe the average behavior of a system of a set of individuals. And okay, and so we can then take our reaction model that's been described as these 2 reactions with these 3 different species, and write differential equations based on the rate of each of the 2 reactions in the system. So the time evolution of Si and R are given in terms of the rates of the individual reactions that affect their concentration. So ds dt, the only susceptible individuals are lost through the infection reaction at a rate R1. Infectious individuals are created through this infection process and lost through the recovery process, and then recovered individuals are created by the recovery process in this model. We have the rates given by the rate laws, and we can see that each reaction contributes to the time derivatives of the species that are either consumed or produced by the reaction. And the coefficient that describes the number of individuals either consumed or produced is called the stoichiometry of the reaction. And so the reactions can be characterized in terms of a stoichiometry vector for each reaction, and the full set of reactions can be characterized by a stoichiometry matrix, which I think I have. Yeah, I have on my next slide. Getting ahead of myself, sorry. And then the full mathematical form of these equations can be given from the rate law expressions that we had previously. Here are two. I think I don't remember if I had it on the previous slide, but it's just a first order reaction in which the rate is proportional to the number of infected individuals. Okay, so this is hopefully review for people. This stoichiometry matrix describes then can be used to build up a general mathematical framework for this kind of reaction dynamics, so that the time rate of change of all the species in the system is given by the product of this stoichiometry matrix, in which each column represents a reaction, the number of species either consumed or produced by the reaction, and then times the rates of the individual reactions. And that gives us back our differential equations. So stoichiometry matrix times the reaction fluxes, which are themselves a function of the concentrations. This is our fundamental equation of reaction network dynamics. And then the formal definition of the stoichiometry matrix in terms of reaction I, and the species I and reaction J. Okay, and I think I covered a little bit already what that means. We don't need to really, the details here are not critical for anything that we're going to be doing today. I just wanted to make sure that the fundamental foundation for how we convert a description of the dynamics in terms of a set of reactions and a set of species get converted to a set of equations that is used to model the system and to track the system dynamics. Okay, and we're going to see two different basic ways that we can formulate the dynamical equations depending on whether we treat the individuals as continuous or stochastic. So whether we have a deterministic method or a stochastic method. I think I said the treating individuals as a stochastic, but I meant discrete if I didn't say that. Okay, so the differential equations, the continuous framework, and there the solution for a given initial condition, the solution is always the same. So we call that deterministic. When we come to stochastic dynamics, then each time evolution, each iteration, each instance of the system that we simulate can have a different trajectory in time because of fluctuations or noise. Okay. Okay, so most differential equation systems models based on reaction network modeling are solved numerically because it's very hard to solve the nonlinear equations that arise from these exactly when there are more than a few variables involved. And these equations in general are nonlinear, meaning that the rates are a function of more than just the concentrations of each, the sums of concentrations of each of the quantities. I have to turn off I thought I had turned off my Slack, but let's see. Just want to turn it off. Notification. All right, it's great that people are asking questions on the fly. I just can't respond to them all in real time in the Slack. I can't check my Slack messages while I'm giving the talk. All right, so we don't have analytical solutions. We need numerical methods. And furthermore, the dynamics tend to involve multiple different timescales. So proteins diffuse on timescales of microseconds to milliseconds. Chemical reactions can take place on timescales from microseconds up to minutes. And so we can get many orders of magnitude and range of the rate processes that need to be considered in these kinds of models. And that gives rise to the phenomenon known as stiffness that makes solutions to the numerical equations more difficult. Fortunately, there are some very good numerical methods for handling stiff problems, and there are solvers, differential equation solvers that sort of seamlessly handle problems that may or may not be stiff depending on their particular characteristics. So a couple of standard packages that people use to integrate stiff equations are CVODE and LSODA. In BioNetGen, we primarily use the CVODE framework to solve equations, and most of the time we get accurate solutions without really having to strain too hard. Occasionally we have to go in and adjust the tolerances for problems that as they get complex. And usually what happens is problems with the differential equation solver arise in either one of two contexts. One where we have we've set the parameters of the model in such a way that there are many orders of magnitude off of their correct values. Typically that involves putting Avogadro's number in the wrong place in our unit conversions, which can easily result in between 20 and 40 orders of magnitude changes in the numbers that are involved, which can clearly give rise to numerical problems. In another context in which the ODE solvers can become compromised or give poor results is when we're doing parameter estimation or parameter variation. So we're varying the model parameters, such as the rate constants, for example, over many orders of magnitude. And what will happen in those cases sometimes is that you'll hit integration errors with these solvers because the parameters are, create an extraordinary level of stiffness. Okay. But for most of our purposes today, we're just going to treat these numerical solvers as a black box, and they're going to be pretty reliable for us. And usually when they break, it usually means we've made a mistake, as opposed to that there's something wrong with the solver or the model. There's something wrong with the model definition as well. But it's usually the mistake is usually on us and not the solver at this level. And so the methods that we'll use this ODE method within the BioNetGen simulate command to do ODE solving, and we'll see that when we get to the hands-on portion shortly. So very simple example from our SIR model. So if we simulate the model with these rate constant values set, as shown here, and these initial conditions. So we start with 10,000 individuals, one of whom is infected and the rest of whom are susceptible. Then we can see that the outbreak evolves in this way over time. There's some peak at a certain time between 10 and 12 days in the number of infected individuals, and then the outbreak eventually resolves as the pool of susceptible individuals is depleted. And so the full extent of the outbreak can be quantified with from the drop in the number of susceptible individuals from 10,000 at the start of the outbreak to something approaching 2,500 at the end of the outbreak. And so, as I mentioned before, we're modeling a collection of individuals. If we're talking about a cell, we're modeling a collection of proteins and other molecules that are, at their essence, discrete. So they're individuals. They occur in integer numbers, not as distributed on a continuum. And the differential equations that we're using to compute the dynamics of a reaction network model are an approximation that ignore this discreteness. And so when the numbers of individuals are relatively small, or in certain other cases, when the effects of nonlinearity become significant, then the discrete nature of the individuals can no longer be neglected, and then the fluctuations of the system, the noise in the system can no longer be ignored. And we need to simulate dynamics differently. And so, for example, in all M-cell simulations, we're simulating the diffusion of discrete molecules that can interact with each other or not. And so the time evolution that's given by an M-cell trajectory always involves integer numbers of each of the species that's part of the system. So if we want to compare our our reaction network model description from BioNetGen with one in M-cell, in some cases we would not be able to neglect the discrete nature of the molecules, and we need to do a stochastic simulation. So I already mentioned this, average behavior. What's important about this, and the reason that I emphasize this whole business of a reaction network abstraction as our fundamental description, is that the same reaction network description, the same sort of listing of the mechanisms that are involved in the system that are used to generate the differential equations and to generate deterministic trajectories, can also be used to generate stochastic trajectories. The algorithms have been developed to generate stochastic trajectories that exactly sample the true probability distribution of stochastic trajectories. That's a little bit of a subtle point to introduce at this point, but I'll come back to it in just a minute. So I guess the key thing I want to say here, I think the thing that this last point is really driving at, is that the basic methods that we use to compute stochastic trajectories are exact, in the sense that, in exactly the sense that's described here, that although each trajectory that is generated is going to be different, you can think of the true dynamics as being governed by a probability distribution, the probability of each trajectory arising, and the stochastic simulation methods that we use by default exactly sample that probability distribution. And so they can be thought of as being exact, even though they give noisy results. So an example of what stochastic effects might look like in a population model, such as we've described, we see essentially similar dynamics between the deterministic and the stochastic, or the ODE and the stochastic. But we see additional fluctuations on top of that. And we'll see a little bit later on that there can be cases, particularly in these kinds of population dynamics models, where the discrete dynamics diverge completely from the ODE ones. You can imagine that if at some point you happen to have a fluctuation that takes the number of infected individuals down to 0, then there's not going to be any more infections after that point at all. And you could see sort of radically different appearing trajectories than you see with the average trajectory. Okay, and we'll get to investigate this in a little bit more detail.
And one thing I'll interject here is to say when what happens is what Jim just described, where you get this big divergence between the stochastic or the discrete simulation versus the ODE or continuum simulation. What I like to say about that is there's a tendency to think of the word average as meaning or being synonymous with typical, right? But in stochastic simulation, you'll often see that the typical behavior is very different from the average behavior. Meaning there could be lots and lots of instances where you get a behavior that doesn't look anything like the average, right?
Right. And we'll see some cute examples derived from this model a little bit later on. Right. So I describe a little bit this Gillespie algorithm that we use to compute stochastic dynamic. Oops, I went the wrong direction. So the standard method that we use is based on so-called Gillespie's direct method, that we use a slightly modified version of that produces more efficient simulations, but it's still exact. The underlying method that's used in BioNetGen for stochastic simulations is an exact method. And so I just illustrate here a little bit to just give you a flavor for how the algorithm works. If we start a trajectory at a point where we have 90 susceptible individuals and 10 infected individuals, then the rates of the reaction are given as shown here, 16.2 versus 1. So it's much more likely that an individual is going to become infected in the next time step, for example, than that one is going to recover in the next time step. The total rate of all reactions is given by the sum of the rates in this vector. So 17.2 per second or per unit time here. We haven't specified specifically what the time units are. And I apologize for the stray animation that didn't get caught here somehow. And then the key idea of Gillespie algorithm is that the time at which the next reaction event occurs is exponentially distributed. And therefore, if we want to sample it, that exponential distribution, we can use this. We can use a random number generator and sample a number, so-called uniform variate, which has been labeled row 1 here, between 0 and 1, and then computing the negative log of that number between 0 and 1 and dividing it by the propensity will give us the correct time distribution for the next reaction event. So, so we pick that the tau at which the next reaction occurs, and then we have to pick the identity of the next reaction by using the fact that the identity of the next reaction is just the probability that the next reaction is occurs in a particular channel is proportional to its rate. So here we have two different channels, R1 and R2, corresponding to infection or recovery. And recovery is much less likely than infection, as sort of illustrated. And we pick another random number generator, pick another random number on this interval, and wherever that number falls determines the identity of the next reaction. So here we selected an infection event. So we fire one instance of the infection reaction, and that involves decreasing the number of susceptible individuals by 1, increasing the number of infected individuals by 1, and we would then go to, I didn't map it out here, but we would have 89 and 89 and 11 for our x of tau vector. Okay, so this just shows how time evolution of our reaction network system evolves, or it takes place using Gillespie algorithm, and we can invoke this kind of simulation on an arbitrarily complex system using BioNetGen's SSA method. Okay, right, and this got a little bit messed up. Yeah, sometimes, yeah, all right. So the basic workflow that I'm going to be describing in BioNetGen. So BioNetGen is a tool that enables the construction, simulation, and analysis of reaction network models. We put a text file that's written in this BioNetGen language that describes the species and their reactions and the rate parameters into a model file called the BNGL file. That then gets passed to the BioNetGen interpreter, which can perform simulations in either one of two basic modes. What we'll be talking about throughout most of today is using this, what we call the indirect mode. So we start out with indirect approach is the simpler one for the purposes of our workshop and for most simple models. And what indirect means is that before we do a simulation, we turn we have to generate the full reaction network that's provided by our model specification from here. And so this generates a set of species and reactions that are placed in this so-called net file that has the suffix .net. And then that file can be passed to one of the simulators that's associated with BioNetGen. And for today, we'll talk about the ODE-based simulations and also the SSA or stochastic simulation algorithm-based simulations. And then those simulators produce a trajectory of the output in one of two formats, the CDAT and GDAT, and we'll talk about those a little bit more as we go later on. In some cases, and we'll see in the talk a little bit later on, this is my second sort of overview when we get into rule-based modeling. It's not always possible to expand a set of molecular descriptions and interactions of molecules into a finite reaction network. And that can happen for a variety of reasons, one of which is simple formation of polymers, for example. Another is when you have the molecular entities become complex, that there's so many different combinations of them that just enumerating all the reactions becomes impossible. And so in those situations, you can do a simulation of the system using what we call the direct methods, which is a form of kinetic Monte Carlo simulation. And to do that, we use something called the network-free simulator, or NFSim. And it can perform a simulation based on a BioNetGen model specification where it's not possible, would not necessarily even be possible to generate the reaction network, but where we can simulate the dynamics of a set of individuals. And then also generate a trajectory, and we can do various analysis of the results. So this method is sort of akin to the kind of results that you would get if you did a molecular dynamics simulation, for example, where you get a trajectory of the interacting molecules, but you don't necessarily know what all the conformational possibilities are in advance. Okay, and we'll talk about this a little bit more again in the next lecture part of it, the workshop. So, and there's a command line interface. So this is how you run a model, your BNGL model, and we'll talk about this in a little bit. But this BioNetGen command is what you hopefully were able to install using pip from the Python package. So it's a simpler way to install BioNetGen than downloading the code directly yourself from the website. Okay. So this slide is from the past, but I left it into the presentation because up until the last several months, the standard way to interact with BioNetGen models has been through this RuleBender interface. And RuleBender was based on the Eclipse platform, and it provides a standard so-called integrated development environment for BioNetGen models that enables you to collect your models together in a single project folder, edit the individual model files with some context-sensitive highlighting that enables you to easily catch errors and other issues, and then there was some visualization capability and also the ability to run simulations directly from the interface and visualize the outputs of simulations. And so in the last approximately year or so, we've been working on re-implementing the functionality that we had in RuleBender for interactive model building and analysis in a new platform. And the fundamental motivation for that came from the fact that the Eclipse platform is primarily based on Java code, and most modern systems biologists have limited facility with Java, and many of the libraries that we were using that were associated with Eclipse, and also additional libraries that we use for visualization, have really become outdated because they're no longer sort of popular and no longer well maintained. And so the regular maintenance of RuleBender had become really cumbersome. But as you can imagine, the process of replacing a large code base with lots of features that we really like, for example, the contact maps that are shown here, that's taken some time. There's been some pain along the way, and it's still a work in progress. But the good news is I think we have a really robust core functionality for our modeling framework that will hopefully be showing you how to use and demonstrating to good effect today. Okay, so that's the past, and RuleBender is still available. It still functions. It's easy to install and set up and use. There's some good videos as well as slide presentations, and for example, the videos from last year's workshop walk you through a lot of different kinds of modeling scenarios using Rule Bender. But today, as I mentioned, today we're going to focus on a new interface, and that's been an interface that we developed using the popular coding platform called Visual Studio Code, or VS Code, which is a pretty new environment, but it's really become very popular in the programming community. I'm sure that many of you who are here at the workshop have used VS Code. Maybe you even fall in love with VS Code. It's enabled us to develop in a fairly rapid way a lot of the functionality that we had in Rule Bender, but with access to many different, sort of a whole ecosystem of different packages and functionality that we could not create ourselves using the Rule Bender platform. And Sinan Salam, who's a postdoc in my lab, has spent a lot of time in the last year really learning how to use VS Code himself from at the beginning, and learning how to do all the coding behind setting up these interfaces to develop the VS Code extension that we're now using for BioNetGen. And we're still trying to find a great name for BioNetGen extension in VS Code, but for now we're just calling it the BioNetGen extension. So that's what we're going to be demonstrating today. So that, if you remember the previous slide, this looks a whole lot like Rule Bender, except we have a dark background instead of a light background. We have a model explorer, a model editor with context-sensitive highlighting, a terminal that shows the progress of a simulation run or a model execution, and an action button that lets us easily execute the model that's contained in the browser window. And we have developed some nice interactive plotting capabilities for the various files that are associated, that are generated by running simulations and parameter scans in BioNetGen. And we'll be demonstrating a little bit how this works in the next portion. And the other thing that we get, almost seamlessly, and actually this is really how I discovered and decided that, oh, we really have to try out VS Code, was the notebook interface that's provided as part of the VS Code environment. Python notebooks and actually the notebook format is more generic in VS Code than just Python and Jupyter, but enables you to have a notebook format to work with models and to do programmatic manipulation of the models that are developed in BioNetGen. And so we're really just at the beginning phases of implementing workflows based on notebooks for BioNetGen models, and you'll get a flavor of that from the presentation that Sinan gives later today. But it opens up a whole pantheon of possibilities for ways to work with these models, but that are sort of starting from the BioNetGen and the BioNetGen extension, but then branching out very rapidly into a much larger software ecosystem. So that more or less concludes the overview of reaction network modeling. And then we're going to jump into, in the next phase after a short break, the hands-on introduction to actually using this framework ourselves to build up simple reaction network models, and then to run simulations and visualize the results. So I'll stop there and see if there are any questions, and then we'll take a short break and then come back and dive into the hands-on part. Any questions so far? All right, I'm going to go back to the schedule here for 1 sec. I think I was faster than expected, but I know I need to break this up a little bit more for my own voice. I also know that the next section is gonna space out and take a different length of time than we were originally scheduled for. So we may have to break that up for. But why don't we reconvene? I'm just going to make this a very short break and take 5 minutes. I want to reset up my the resolution on my screen so I can hopefully avoid some of the lag effects that I've been having. But so we'll start back up at 9:20 Pacific time. So another 5 minutes or so. Okay, I'll be right back. Okay, I am back. I was just checking out that a few issues are, people are having some issues with VS Code that I see some discussion of in the BioNetGen channel. Hopefully, people will be able to get that resolved. It's not going to be critical to follow along with everything that I do today interactively in your own terminal, but if you can, that's great. But if not, that's certainly not the end of the world. And I'm not sharing yet. Okay, good. So if you're continuing to have some problems with the installation, definitely work with us, and Sanan in particular will be helping you. We have some other people available as well from my group if you run into problems along the way. So we should be able to get people help as you guys need. So don't panic if it's not working for you. And you know, and if you're not able to get it working as we're going along, just follow along with what I'm doing, and I think you'll see, you know, you'll get the basic ideas of how things are working. I've tried to lay out the steps very systematically in the slides. You should be able to go back through and work through it yourself at any point in time in the future. Yeah. And I see that, yeah, we will also put links on the in the Google Doc. Apologize a little bit for that. We're a little bit disorganized. There were so many moving parts for this for the workshop this year that we got a little bit behind. But I think we will have all the materials ready for people when they need them. Okay, so let us go back to the my screen here and the PowerPoint. Here we go. And hopefully you can see Flagman. So this is just the homepage for the VS Code BioNetGen extension and the instructions, which I think many of you already tried, and hopefully most of you got to work. And if not, it will be pretty straightforward. All of the tools that we're using here and were chosen because they're widely used and easily accessible. Problem is that on modern computers, there's so many different possible configurations of all these different tools, and sometimes they don't play well together, that you can hit glitches. Okay, so I have some slides here that introduce launching the VS Code for the first time. I think I had meant to even start with a VS Code not initialized. So I'm going to be for this presentation switching back and forth between the Visual Studio Code and the PowerPoint slides. So hopefully that will work out pretty well. We'll have to see. I remember doing it last year. We had a few hiccups. We'll see how it goes. Okay, so so when you bring up VS Code, you should see a welcome window that looks like this, and you guys are seeing my Visual Code VS Code screen now. Are you seeing both? Are you seeing the PowerPoint slides in the background as well?
Yes, we're seeing both.
Okay, so we can see, you know, the real thing and the picture of the thing at the same time. I might be, at some point, if that's distracting, I can just pull this and make it bigger too. So the key thing is to make sure that you have the VS Code extension installed for BioNetGen. And so the extensions are managed. So first, welcome to VS Code. This is a window that, and the basic home window gives a lot of shortcuts for doing things that you might want to do, like just opening up projects that you previously had open. If I fumble around from time to time using VS Code to do some stupid things, I expect I will along the way, because I haven't been using this for that long, and I'm still learning some of the stuff. So I'm sure some of you guys will be much more expert at dealing with at VS Code than I am. So bear with me, and also feel free to jump in if you see me doing something that could be done in a better way. Feel free to jump in and let us know. Okay. But as far as looking for the extensions, we want to make sure that the BioNetGen language extension is installed, and we can see here that it already is. We have version 0.4.8, which is already a more recent one than the last one I remember using. So it's been updated. A lot of information, basic information down here, and so on. And there are tons and tons and tons of extensions that are available for VS Code that help you to work with all the different kinds of programming tools and associated language formats. So we developed one for BioNetGen, and we'll get to play with it now. So we shouldn't have to mess with the extensions much more. I guess one warning is that we want to make sure that the dark mode is selected as the color theme. That's the one we have that currently works. Okay, so if we go back to our Explorer window here, the first thing that we're going to have to do is open a folder. So let me just follow along the instructions here. So I navigate, I showed you the extensions, and then we have to open a folder where project is going to be stored. And here what we're going to do is create a new one. And I call this one session reaction net. So we open a folder, and I'm just going to put a random one on my Dropbox. We'll call it that, and click open. And now we have a project folder that's empty. We still have our welcome window open, but I can close that. And now we just have a completely blank interface here. And let's see what we're going to do next. So the first thing that we want to do is make a model, a file for our model, for our SIR model. And we do that by clicking on this new file icon that's in the Explorer next to the folder name, and we name it SIR.bngl or whatever you want. The key thing is that in order for the language extension to recognize that it's a BioNetGen language file, it has to have that BNGL extension associated with it. So right, let's do those steps then. So we go up here. If you're looking at your window, you'll see that there's nothing. There are no icons displayed. They only show up when you mouse over. I'm sure that's something that it's a feature that I could turn on or off. Click that, select a new file, and let's call it sir.bngl. I'll call it in all caps this time just to differentiate it from our example. And now I have the BNGL model file. Now, it's an empty file, and it's a language that most of you haven't programmed in before. And so it's a little bit intimidating. Okay, well, now what do I do? And one of the things that is really great about VS Code is a feature that are called that's called snippets. And we'll see the power of that in just one sec. There's one thing that I meant to tell you to do, which is to make sure that the autosave is turned on when you run this. You're welcome to run it, to do it without autosave. I love autosave because then I never forget to save the model. And so inevitably, if you don't turn on autosave, and then you go over here to click execute the model, then whatever change you last made that you forgot to save, well, it's not gonna go into effect. So I like to avoid that problem from the get-go by having autosave. Plus, it makes me feel modern. So we have autosave turned on, and then yeah. So the basic structure of the BioNetGen model is shown here in this slide. It's a series of blocks that are encompassed by begin and end commands. So begin model and end model represent the full scope of the BioNetGen model description, and then the elements of a model that can be defined parameters, seed species, observables, reaction rules. There's some additional elements that we'll cover that are more advanced later on. And so this is our model and the actions. And in the past, you had to kind of remember these, the names of these things and, what they meant. But with snippets, we don't really have to, get into the details of the syntax. We can get a template for us automatically, and we'll jump into that now. So each of these model elements has a function, and we'll figure those out as we go along. Just put this up. Okay. And okay, so the snippets feel the power of the dark side. So it really is, kind of a brave new world of programming using snippets. For those of you who are used to it, it's just, just standard, but it really was a revelation to be able to do this stuff. So if we hit B, then we get a menu that where it provides all of the different options that the extension knows about for model elements. And so the easiest one, the one that we want to get the full template, is the begin model. So if we have a begin model, that will encapsulate all the different kinds of blocks that can, components that can occur in the BioNetGen model. So we saw here, we type B to begin, we select begin model, and then the dark magic happens. And we have all the model elements. And yeah, so for the purposes of this demo, I don't know, Zoom is great in that people can adjust their screens as they need to be able to see, but it's usually good to make the text a little bigger, just in case. And so you can adjust the text size by using command plus and minus, which is what I was doing here. Plus makes it bigger, minus makes it smaller. And that's very useful, especially for those of you who are on the older side. Your near vision starts to break down, and sometimes it's really helpful to make things bigger. All right, so then we can jump in, and now we know that we have to fill out at least some subset of these model elements in order to make the model. So if we go into each block for parameters, we can type P, and we see there's a number of options. The one that's relevant for us and will be relevant in any of these blocks is the one that's a block name and followed by a line, so parameter line. And then bring up one line where a parameter's name and value specified. So this is like a form, and we can type text here, and I have to remember exactly what I was supposed to type. Oh yeah, okay. So the first parameter we want to define is n. So we get our parameter name, we type n, and then the value to go to the next element in the form, we type tab, and then we type in the value. So that looks like this: capital N, tab 100, and now we've defined the first parameter of our model. It's useful to hit tab again to get out of the form, and then we hit return and go to the next line. It looks like when I did that, when I was making the slides here, I actually didn't do that. You can see that it's still highlighted here. That will cause the snippets not to work until you exit out of that particular form. So then we can repeat for the remaining parameters. So we have I0, R0, and then G. And oh, I wonder, I left this out of the slides where we define this basic reproductive number, which is the number of individuals that get infected on average by a single susceptible individual. And that can be defined in terms of the other parameters of the model, the fundamental rate parameters, G and beta. G is the lifetime of the infected individual, the time constant for the recovery of from infection, and beta is the infection rate. And what we did in setting up this model was to use R0 is a base parameter, so a number that a parameter that we gave an explicit number to, and then I defined the infection rate in terms of the other parameters G, R0, and N. So it has that relationship that's shown here, and that illustrates a key concept of the parameter block. Basically, the other main syntactic element is that besides names and values, the parameter values can also be expressions of other parameters. So any sort of basic math expression, and also basic math functions, elementary functions of parameters can be used in the parameters block. So here we just defined it using this formula for beta. So we could go ahead and do that. So we have I0, 1, we have R0, and I have to remember 2.5, beta, sorry, it was gamma 0.33. And I could have used parameter line for each one of these as well, if I forgot. Parameters are pretty easy, but beta, and then we can do beta is equal to gamma times R0 divided by N. Okay, so parameters are pretty straightforward. I already talked about that, and any parameter, maybe an expression, we can use basic math functions. Scientific notation can be used. So here I'm going to change 100 to 1.0 E2. Those are equivalent, and that's it for the parameters block. So this just enabled me to set up the model, the parameters that I'm going to use to govern the initial conditions and the rate constants for my model. We're gonna skip for this first introductory model the molecule types and the compartments, but the molecule types, these are going to be used to define structured species, which are really a core concept in BioNetGen, that the molecular species have properties, and so they can have elements that interact with other molecules. And when we have this kind of structure, so that an individual molecule may have binding sites, for example, or sites that are modified through phosphorylation and so forth, we'll define those elements in this molecule types block. But for now, we're just going to skip that. And then compartments define, the spatial topology of a system, and we'll use those to make models that have explicit spatial topology that then can be transferred to MCell directly and used for simulations. And then finally, functions enable us to define variables that are time-dependent functions of various concentrations and properties that are defined in terms of concentrations in the system, functions of observables. And we'll do that again later on. They're used basically to set up non-mass action rate laws that I mentioned. And for the context of what we want to do with MCell, that's not currently so useful because we can't use those functions directly in an MCell simulation just yet. We're working on it. Okay, so we'll skip all of that. And so the things that we're left with having to define are the fundamental species. And we'll talk in just a sec about exactly what we have to do, the work we have to do there. And then observables. Why did that come back? Okay. Observables, which are going to define the outputs that we want to directly extract from our simulation. And the observables are the quantities that will directly result in an output file that will be plotted when we execute the model in the VS Code extension. And then really where the action of the model is in this reaction rules, defining the basic interactions that can take place in the model, how species are transformed from one to another. So, okay, so the first block that we have to grapple with now then is the species, which are used to define species that are initially present in the system and their initial concentrations. So the basic steps for adding species are a lot like what we did for parameters. We make a new line and indent 2 spaces. That's the custom that I like to follow. It's not required. And then you type SP and the magic snippet appears and you hit, you get species line, and then you'll get a species line template. So go and do that. First, we get rid of the molecule types and compartments. We hit return, space, space, species line. And then the first species we're going to define is the susceptible species, so S. And then the convention, it's not strictly required, but the convention we're going to use here is that all species names have parentheses after them, but we're going to leave those empty. So for the basic reaction network modeling that we're doing now, we're building up our model based on species that are identified solely by their name. So here, S defines the population of susceptible individuals. They don't have any further specification than that. There's not susceptible individuals who are, have, I don't know, some attribute associated with susceptible individuals, say male, female, for example. So there's only one type of susceptible individual, and we're not further distinguishing. So these are so-called unstructured or flat species. And we denote that with the open parentheses. Then if we hit tab, we go to the species amount, and we need to pick either a number or parameter here. And just like with parameter values, the value of the initial amount of the species can be an expression. So here, we can say that the initial number of susceptible individuals is the total number of individuals minus the number of infected individuals. And so it's a little bit dangerous because we don't, n in I0 could be such that the initial number of species is less than 0, which would not make sense. But here we've defined them such that that is not the case, so it's okay. And that's good for S. And then the other initial component then of our system is going to be the infected individuals. And we know that those are initially present. I tried to tab when I shouldn't have, as I0. So they're I0 infected individuals initially. And so what these species, what this species block is declaring is the species that are present at the time that I'm initially going to begin simulation or any other operation on my system. So I'm setting up the initial configuration, and going back to the lecture notes, it was the values of the elements of that X vector. That's what I'm declaring here. The interesting thing about the way that BioNetGen works is that we don't have to initially specify the number of recovered individuals. The network, the process of network generation, when the system evaluates the rules that we'll see below, that will automatically find all the possible species. It says, there is a question, why should we not tab? Oh, use tab instead of choosing. I'm not 100% sure about what's being asked there.
Can you hear me?
Yes, I can hear you.
So I've been typing away as well, and instead of when I get into begin species, instead of typing species pattern and choosing the species pattern option, I just hit tab and I write the name of the species.
Yeah,
but that's yeah.
for this one, you should choose species line. If you choose, oh yeah, okay, so species pattern.
Yeah.
You can, so you don't have to do that. The snippet is completely optional. Is that what you're asking basically? I mean, I can just type also. As long as I know the syntax, it's great.
Okay, okay. Just wondering if something
Yeah,
would go wrong.
Not required at all. It's just a helper. It's just to help you, you know, it's so that you don't have to remember all the mnemonics when you're learning the language. That's a big thing. And it can get more complicated, as we'll see. There's going to be some more. It's especially true when we get to the actions, because they tend to have a lot of options, and you can't necessarily remember off the top of your head what they all are. And it's better than having a cheat sheet that has it all written out for you. We do have such documentation, but this really helps to minimize your dependence on that starting out. So, okay, so this was our species block. I think I did actually put the parentheses in. Later on, I forgot the parentheses. And this is all something weird there. We did this, and now, so we have we have expressions. The initial amounts must be greater than or equal to 0. It's permissible to put R as initial species and give it 0 concentration, but it's just not necessary. That's why I left it off here. That's the next comment, and then a statement about units. So for the examples that we're describing here, we're going to use the molecule counts as the basic units, assuming that we wanted to make a simulation that we can simulate either stochastically or deterministically without changing the parameter values. So we strongly recommend for that reason using numbers or molecule numbers. Here, they're obviously we're not meaning them to be molecules. They're meant to be individuals, organisms that are in a population, and it becomes mandatory to use these molecule units when we want to set up our model so that it can be passed directly to MCEL4 without further manipulation. I think that's correct. People have to correct me if I'm wrong about that. Okay, so we're about halfway there now. We have 2 more blocks to define. This block is also sometimes called seed species, and the seed, I just mention it here because the idea is that the amounts that we give associated with the species are the amounts that are initially present, and the reason that we call these species collection as a whole seed is that when we go to generate our reaction network from the rules, we're going to start with this collection that's defined in the species block. And you'll see that here, when we do the network generation, we'll expand out to get the recovered species, so the R species. But in more complex rule-based models, often the seed species might be a handful of different molecules, and the reaction networks that's generated may have hundreds of species or even more. So that's just a little bit of background. So the other thing that we need it's not mandatory to get output from a simulation, but most often, especially for rule-based models, we're going to want to define observables. And these are the outputs of the model that are going to be put in this file called the GDAT file that's produced by a simulation. And like I said, in the context of a rule-based model, that's going to be the primary way that we get information about the simulation, because the individual species that are part of the network, sort of, you can think of them as having very fine grains of detail that are not directly experimentally measurable. But the observables represent sums of concentrations over species that share a particular attribute. And I'll go into more detail about this later on, but I just want to set the stage that the idea is that the observables are going to be used to relate detailed properties of the model species to measurable properties of the system. Okay. And so we have back our window again, and the process for adding observables is similar. If we indent 2 lines and type O, we're going to get snippet for the observable line that we can select. And then something interesting is going to happen when we do that, and that is that observables now have one of the elements, the first element of an observable is a type that can take on 2 specific values. There are 2 basic types of observables. One is called molecules and one is called species. And the types are distinguished by the way that the way that the observable is computed as a sum over the species that have a certain property. And we'll talk about that a little bit more later on. But the sort of your default selection should be molecules observables. But when you're dealing with structured species, then sometimes it makes sense to use the species observable. And I think I'll just leave it at that for now. So we'll pick molecules for this example. It will not make a difference which one you pick in this case. And then tab, and then, so in this model is very simple. We only have 3 species. And so we're only going to, all of our observables are going to correspond exactly to a single species. So it's rather dull here, and you might even call it tedious. But we're going to generate species observables for each of the possible species. And in this case, we'll actually generate an observable in the last line, line 19 here, for a species that was not specified in our species list, because that we happen to know is important, the recovered individuals. Okay. So that's what we do. So we'll go back to our window and just do it quickly, just for repetition purposes. Yeah, tab's going to put 4 in. So we have observable line, select observable line, select molecules, and first species observable name is going to be S. I'm not going to put parentheses after that because this is actually a name, an observable that we don't typically put parentheses after. And then the species will have parentheses. And I'm going to do that for, I can do it free form. We can see what that looks like. You see that the highlighting changes as the grammar detects the necessary syntactic elements. And I still almost automatically want to hit save after each time I do these things. Okay, so now we have our observables block, and we're ready for the prime time action, as it were, actually defining what happens in our model. So the reaction rules, and that where the action is defined. So the basic syntax of the reaction rule is given here, and very fortunately, you don't have to memorize this at all because the snippet will generate it for you. It might, for some reason, my mouse is very sensitive, and if I swipe it just the wrong way, it will advance the slide. So the key idea of a rule is just to remember is that it's just chemical notation with an additional name modifier that can go at the beginning. So reactants to products and a rate constant. If the arrow is one-sided, it's just a unidirectional irreversible reaction. And if the arrow is double-sided, then it's reversible. And for a reversible reaction, I have to have both a forward rate constant and a reverse rate constant. And that's pretty much it. And as I said, fortunately, the snippet will remember that, so we can go into our reaction rules block, and the abbreviation that most efficiently picks out the rule line is RU. So you type RU and you get a rule line, and it looks like that. It got a little bit cut off here because it's kind of long. And then we can tab through our rule. We can name the rule R1, and encode the reaction S plus I goes to I plus I with rate constant B by tabbing through. And so that would look like space RU line, and then R1 S plus I, and I'm going to go back and put the parentheses in. Oops. And then I lost my form. I don't know how to get it back. I don't know how to get back into the form if there's a way to do that, but it's not too hard to just to replace the text. And that's wrong. So I need to fix it. Oops. Sinan, do you know if there's a way to get back to the form?
Not that I'm aware of.
Yeah. B. And sometimes the snippets will get kind of overeager and want to put in model elements when all you really meant was B, and you could just ignore it at that point. Okay. And then we need to do R2. I'll see if I can get through this one. And R2 is just going to be
and then it be oh sorry, no it is.
I'm sorry?
Nothing, I was getting confused with the G and B, but it's okay.
Yeah, it is a little, it's confusing because G is defined before B.
Yeah, it's fine.
Yeah. I just wanted to go back and enter it directly. So then I don't know what the most efficient way to clean this up is. You can tab through. Oh, and now I messed it. Yeah, that is actually good. All right. It did go back in somehow. Anyway. There may be people who are more snippets experts than we are. It has a little bit of a mind of its own sometimes, but there we go. You know, in the back of your mind, you should always remember that it is just a text editor, even though there's all this other stuff that can pop up from time to time. Okay, so now we have our reaction rules defined, and so we have our complete model specification. And now we need to do something with it so that we can actually see the results of running a simulation. So to do that, we need to define actions. So here I went through the, so we have now our full model. And now actions, this is where the real action is. This is when we actually do something with the model itself. So these go after the end model line. And as shown, and the key actions that we're going to deal with from the beginning are generate network and simulate. It turns out that the generate network is actually optional. BioNetGen will automatically figure out that it needs a network if you pick one of the simulation methods that requires it. But there are contexts in which it's nice to have the generate network command explicitly, especially if you're going to export a model. So it's not bad to use it, to have it. Right. Thanks for the comments. Start with a hash sign, and this was our first actual example of it. I swear PowerPoint makes me crazy because my cursor has completely disappeared. Okay, there it is. Right. So we see first example of a comment. The comment can go anywhere on a line. You can the appearance of the hash symbol means that the rest of the line is commented out, like just as in Perl or Python. Simulate command. Okay, so these are the two commands that we're going to be working with. For generate network, we just need G, and then we select the overwrite option. That's the main reason I wanted to put in the generate network here. If you run BioNetGen from the command line as opposed to using it in the interface, it's not a bad idea to have the overwrite set because, excuse me, it will fit if you run when you try to run the model twice. The second time it will stop if you already have the net file having been generated. So and the overwrite option prevents that from happening. And then we can add the simulate command using also a snippet. So I'll just go through and show you what this is going to look like before we actually do it. And you see the different methods that are available. We select ODE. And then this is where I think the snippets can be really helpful, especially when you're getting started. It automatically shows you what additional parameters are needed for the command in order to make it, so these are the minimal ones, the ones that have to be set in order for the command to be carried out. There are many additional parameters that are associated with simulate that we will take advantage of as we go along. And I'll show you where that's documented as well. So here we're just going to set the end time. So the simulation is assumed to start, the current time is assumed to be 0, and the simulation time, the ending time for this simulation is set to 30. And we do 200, we do that simulation for 200 steps. So that means that between 0 and 30, they're going to be 200 output points. If you want to, if we wanted to, for example, make the time spacing equally spaced, if we wanted to, output the trajectory value at each integer time from 1 to 30, we would need to select 31 steps in order to do that. Okay, so that's what that looks like, and we're ready to go. Once we have that entered, we can click on Flagman and run the simulation. So, so we go G, we generate our network, we select 1. Randomly hit tab, and then we do simulate. We select the method is ODE, T-end, then we said was 30 and 200 steps. I just like to, and the number is arbitrary, but I just pick a largish number to make the trajectory look smooth. And now we can just hit run, and hopefully everything just works. And okay, so we get this, we get the output as a GDAT file. And what VS Code is going to display is just the text file. It turns out to make it launch graphics without displaying the text file first is either extremely complicated to achieve or not possible, at least at the moment. But fortunately, so it's actually a feature because we now have a basic text-based output that you can see, and you see that the numerical simulation data is just recorded in a simple tabular file. You would realize very easily that it's very easy to import this into a spreadsheet or to import it into MATLAB or Python or whatever tool you want. There's lots of tabular data is very easy to work with in this form. And sometimes it's even worthwhile you can debug simulation results by just looking at the values that are in the file. More often than not, we would like to make a plot. And so there are a couple of different tools that we can use to make plots in the VS Code interface. The one that I recommend for interactive work is the built-in plotting. It's the first one shown here. And that makes a very nice interactive plot window that's using Plotly. So we get Plotly plots of our GDAT files. And this has a lot of really nice features that we can briefly mention. And I made a list for myself of what to talk about to keep myself focused. So lots of things happen when you execute the model. I mentioned the plot stuff, but there's actually other things that happen. So the first thing to notice is that a bunch of files got created. So if we go back to our VS Code window, this one is simpler. So we've only done, we made our model sir.bngl, that's in the top level directory. And when we run run the simulation, the extension automatically generates a subfolder that's time-stamped. It's in the top level directory called SIR, the base of our model name here. So the SIR model, and then it's time-stamped to the exact year, month, date, and then time when the simulation was run. And that's these timestamp directories, each time you run a simulation, will be created and they'll appear in the order so that you have a complete log of everything that you've done. At some point, you might want to clean that up and remove older ones that aren't useful, but by default, they'll just keep being generated. And then all of the files that were resulted from executing that simulation file are in this subfolder. So a copy of the BNGL model is saved there because that's the model that was used to actually generate the output in this directory. So if you wanted to rerun exactly what was here or see exactly what the parameters were, what simulation commands were used, whatever, that's in this the BNGL model. So this is just a copy of the model from the time when it was executed. And then the output files are the CDAT and the GDAT. We didn't talk about the CDAT in detail yet, but we will look at that in just a sec. And the GDAT, of course, is what we already plotted. It shows the observables as a function of time. And the net file, which we'll look at a little bit later, shows the reaction network that was generated at by that generate network command that we issued. And this can be sometimes helpful to look at this file to see, especially when we have structured molecules, when we're building a rule-based, a more complex rule-based model, we can actually look at what happened during that network generation phase by looking at the network file. And we'll go into that a little bit later on. Okay, so going back to here, I want to follow my script. Right. And okay, so I mentioned the files that are generated in the Explorer and the GDAT file. So the GDAT file is automatically opened by the simulation action or by the simulation button, and then we press the plotting button in order to make our plotly plot appear. And then the last thing that I haven't mentioned so far is that the output of running the model with this model file is displayed in the terminal. And it's very useful to look at this model output, especially when it didn't run, because any errors, any syntax errors that I had in my BNGL model will be displayed in this terminal output. So just to give a brief example of that, like if I had, if I forgot that the parameter names were, for example, case sensitive, and I wrote an expression like this and tried to execute the model, I would get this reasonably helpful error message that says, I can't find reference undefined parameter lowercase n at line 12. And I go to line 12 and I see, oh, it's lowercase and maybe I should use an uppercase n. So that's an example of looking for the output in the terminal window. It's a good idea, especially as you're developing a model first, when you're starting out and running it for the first time, to actually look at what's going on here. And we'll go back to examples of this a little bit later on. That is the main thing. So I mentioned, so the next slide covers interactive plotting. And there's lots of features of the Plotly plots, some of which I can leave you to discover on your own, but we can look at a few of those. So it's very easy to double click one item here in the legend and get then a plot only of that item. So, and it'll auto scale to that species or that observable. So that can be very helpful. You can also zoom in, and this in my layout now, the plot is a little bit cramped. These extra buttons up at the top sometimes impinge on the items that are in the window, but you can resize to avoid problems like that. You can see that this I don't know, tooltip, whatever you call it, is displaying the values of the individual points that are in the trajectory. We can actually display those explicitly by showing the markers. So, and these are just the points at which the simulation was output. I should mention so you hear it at least once, that the accuracy of the numerical integration does not depend on the number of time points that are chosen for the output. So if I display 200 points or 20 points or 10 points, the actual values of the observable, of the solution at those points will be the same in each case, or very, very close to the same in each case, because the accuracy of the solver is set internally and does not depend explicitly on the output points. Just something to keep in mind for later on when you're working with your own models. Okay, so we can turn the legend on and off. We can also log scale the axes, which is kind of a weird thing to do in this case because it's time, but in some cases that might not be so strange. So we could log scale X and we can log scale Y. Oops. Which can often be useful. Plotly has its own kind of interesting way of displaying log scale, which might take some getting used to, but it's actually pretty nice once you're used to it. So some other useful features up here. The main thing, auto scale, we'll use that in some of the examples that I'll show. You can also export the plots that are shown in the window either as images, PNG format, or SVG format just by clicking a button. So if I click the button, then you see that a PNG was added to the folder where the simulation ran. And actually, one of the beautiful things about VS Code is that we'll display many different kinds of file formats either automatically or by installing the right extension. And in this case, I don't remember if the PNG is natively supported or whether I installed an extension that supports PNG display, but there it goes. The similar thing happens if I export to SVG. I get an SVG file. That one, you know what? And I'm surprised that it didn't happen. I haven't installed the extension on this particular computer for the SVG output. So like what I can do in this case, if I go to the extensions marketplace and type SVG, there are going to be any number of different extensions that will display SVG, and I can download that by clicking on it. And I think if I go, oh, that's my model. If I click on that file again, I get this extra button now that will let me preview the SVG. So it's very flexible. Okay, other things about the plot. So we can also do interactive plotting of the CDAT file. So I haven't said what the CDAT file is. Let's look at it for a sec. It's another text-based tabular format that here it displays the time courses of all the individual species that are in my model. So here it's exactly the same as the GDAT file, except that the headings instead of saying just S1, S2 through SN in the CDAT file, because the species are just named by their index. In the GDAT file, they correspond to the observable names. So here in the CDAT file, the columns are headed by the species index. But the plot is going to be exactly the same. Good. Right. Yeah. So that's a general thing about VS Code, that the extensions will provide functionality for interacting with whatever is in the editor window. And so Flagman is connected to the run BNG function, and it basically does a BioNetGen run -i file name with some extra options passed to it to put all the output in a single directory that has a certain name. Okay, and so we have our CDAT file, looks a whole lot like the GDAT output, but there could be hundreds of time courses in the CDAT file. Again, sometimes it's useful for debugging. You might see a situation where only one of the species is populated, for example, or species concentration is going off to infinity, or increasing linearly in time. All of these can be diagnostics of problems with your model. So we'll get rid of that, and that one. And I think, yeah, I showed the plot there. Okay, so let me just stop there for a moment. And the next thing we're going to do is show that, how we can manipulate the model a little bit and do some simple investigations of model properties and how the model depends on the parameters, and also how to do a parameter scan to systematically investigate model dependency on parameters. But before I do that, are there any questions that people want to throw at me? Everyone is quiet. I feel very, okay, I see there are 58 participants. Well, that's great. So people mostly.
Question?
Yeah.
So what other applications can we use this calculation for in general?
So basically any kind of system that involves, that you can, any kind of dynamical system that you can represent using this reaction network formalism can be modeled using these tools. And it's almost hard to answer the question, basically anything you want. And what we're going to focus on through most of the workshop is molecular description. So modeling things like ligand receptor binding to, an extracellular ligand binding to a cell surface receptor, the dynamics that, surface receptors can undergo, both, reacting with other receptors on the surface, or binding to molecules in the cytoplasm of a cell to form complexes, and then to trigger other cellular events. So, yeah, really any kind of biochemical system we can model very easily using these tools.
I saw a trajectory file when I was installing the Blender, and it was a PDB trajectory file. And I'm curious, are we able to get trajectory files for this, like data, as in protein PDB?
Do you want to answer that question, Tom? You're muted.
Yeah, sorry. I'm not exactly sure what you're asking. So PDB is the protein data bank, and it contains, I'm not super familiar with it because it's at the atomic molecular level.
Actually, so what I was asking was like, I'm sorry for cutting you off, but I was trying to find out whether the data that we got from the calculation can be kind of converted to a PDB file.
PDB, protein data bank.
Yes.
Protein data bank. My familiarity with the protein data bank is limited to atomic structure of protein. And the calculation that was just done is just the system of differential equations describing the interactions of these molecules as populations of molecules. And the graphs are graphs of the numbers or concentrations of particles in the population of the different populations. And that information is at a much coarser scale than the distributions of the atoms within the proteins. So I'm not exactly sure what your question is about.
Okay, I think I understand now. I was getting it a little bit confused.
Okay.
Yeah, thanks, Tom. I sorry to send you. I was just confused because of the cell blender connection, so I thought that might greater insight.
So maybe, maybe to say just a little bit more about it. So when we simulate the systems like this in MCell, which we'll do tomorrow, systems like this, rather than treating the particles as belonging to a whole population, as you've seen here with the ODEs, we treat them as individual discrete particles that actually have discrete identities and locations in 3D space. But still, those are just particles representing each species and not the actual structure of the proteins that they represent. So it's still a coarse-grained representation, even when we look at it in MCell.
Okay, got you.
Yeah. And tomorrow you'll see that we can represent those entities with shapes, glyphs that are purely, right now, purely just graphical representations, abstractions, just for visualization purposes and not for real protein structure, protein docking, that kind of interactions between the binding sites, ligands, and their binding sites. We don't go all the way down to that very, very fine scale with our tools. Okay.
Let me just jump into, there's some question in the chat that's very useful. So example models, I definitely, there's lots of example models, and I posted a link there, and I'll highlight it in just a sec, to the BNG tutorial site that has a whole bunch of tutorial examples that are annotated and give you a lot of different, a view of a lot of different features and use scenarios. There are many, many more than are there, but that's a good place to get started. There are also models that are part of the BioNetGen distribution that you can find. Yeah, someone posted biomodels.net. We can, BioNetGen can actually read SBML models, and it has a tool that can convert the models in biomodels.net, which are in this SBML format, into structured models. But that is not currently included in the distribution that you guys have through PyBioNetGen. We're working on getting that back and integrated. So that will be coming soon. But you can import SBML models into BioNetGen using that atomizer tool, and Sinan can provide more details about the best way to access that at the current time. Right, I think some of the confusion about and things that people are asking me about demonstrating like the seed species, that some of these things will be addressed as we get more into modeling structured molecules in BioNetGen, and you'll see why we really need to use network generation for cases where we have a lot of complexity in our model species space. But let me just, in the interim, I have to share my screen again. Hang on one sec. Okay, so the link that I highlighted in the chat there, small, was to this BioNetGen tutorial page. I actually started this tutorial for a previous workshop because I wanted to have a whole bunch of examples. And in the first workshop, I think it was 2017 or 2018 that we did this. I tried to go through all the examples. I had the idea I was going to go through all the examples that are in this tutorial in my presentations over the course of the day. And that did not happen, which will not surprise you if you try to go through this. But so there's a lot of models and a lot of richness that's embedded here. And I tried to highlight for each of the examples what you can hope to learn, what features are demonstrated by each of the examples. And we will use a few of these as we go along. I guess, the SIR model is here. So the model that we're using, a version of the model we're using in the workshop this time is here. I parameterized it a little bit differently. I can see that right off the bat. And that's the main difference I see. But so, and all you have to do, for example, to investigate this model, the easiest way to, you can fork the repo or clone the repo rather, and directly within VS Code. I will show how to, maybe I should show how to do that a little bit down the road. But all I have to do just to take one model is to create a new file, call it sir_old.bngl, and then paste in what I had, and voila, I could see exactly what was in this model. And the highlighting is not happy because it doesn't have parentheses here. Put that in and fix it. And then this is the old, the one that I used in a previous workshop. I can simulate it and see what it does. I ran for a longer time. I can see that right away too. I think in this model, I did a stochastic simulation along the way. Whoa, oh, and this one's crazy because I think for these parameters, yeah, for these parameters, the infection just dies out. This is a good case, right? The infected individual didn't pass it along. We will do this in just a few minutes. But so to summarize, this BNG tutorial site provides a wealth of information. We will draw some models. We'll cover some models from this in the activities today, but there's way more here than I can show you. So the key is that this is a repository. So if you bring it up in your browser, the stuff at the top shows the actual file structure, but the README is the thing that has all the information about how to use it. Okay, this was originally constructed for RuleBender, and we haven't. In the video, we'll demonstrate how to use it in RuleBender, but all the models can be used just the same. So this was an even simpler introductory model that illustrates just a simple binding reaction. So, or a bimolecular reaction where A plus B goes to C, that can be used to represent the binding of two molecules in solution. So that's one of the simplest models you can make in BioNetGen. So we'll come back to this site again over the course of the workshop. But thanks for asking that question and reminding me that it's very useful to have access to all the code. We are putting all these models in the ones that I'm talking about in the presentations, and also for Sanan's presentation later as well. We're putting those in the workshop repository, which the link was already put up for that. In the BioNetGen subfolder, you'll find all the BNGL models. Okay, did I hit everything in the chat, or is there anything else that I should? Sometimes. Right. Okay, I'll come back. We'll talk about competitive binding down the road. But let's come back to our SIR model, because we have some, and I think some of the questions will be addressed by some of the scenarios that I have planned out. Just, it's really basic stuff. I think the goal here is to introduce some of the core concepts to people who are not as familiar with it, and to people who know those concepts well and know these workflows, show how to implement, how to carry those out within this tool. Okay, so we can investigate the effect of changing the parameter in the simulation by just simply changing a parameter and rerunning the model. So that's very simple to do. It's shown here. So here I just go to R0, change it from 2.5 to 1.1, rerun the simulation, and we get a new GDAT file, and we see a new time course. Okay, and it's obviously very different from the one that I got before, which showed a peak and then showed the outbreak, the infection level resolving and going back down to 0. Here, on a timescale of 30 days, the number of infected individuals looks like it's still rising, and because susceptible is still going down and recovered is still going up. We can zoom in. Yeah. If I double clicked on the number of infected individuals, we could zoom in just on the infected time course. We'll do that when we get back to VS Code. But if we wanted to visually compare the trajectory, the outputs from the previous simulation to the current one, it's very easy to do that in VS Code by splitting the window, splitting the editor window so that we have side-by-side view, or we could do split it vertically as well, but here I'll show the side-by-side one. So if I click on this split window item here, it splits the window, and then I can just drag one of my plots over into the new window, and it won't look like this immediately because I have to auto size. I have to hit the auto scale button, which is shown here on this window, on both in order to get them to fit properly in the newly resized windows, but it's pretty easy to do that, and I'll show, we can walk through that. So this was our original model. It's also worth mentioning, so we ran this model twice. Oh, the second time with a syntax error, so it didn't generate any output. I'm just gonna actually delete this one. And then, so we ran the model twice. I had closed the GDAT window, but the GDAT file is still there. So I find if I click on it, open it again, in our text format, but then I just click on the plot window to open the plot. And now I have my plot of my original model run. And so I can leave that open, go back to my editor, change the parameter value for R0 from 2.5 to 1.1, I think I said. Is that right? 1.1? 1.1 or 1.2, can't remember. It's well past lunch in East Coast time too, which does raise the point that we should find a stopping point, perhaps. Okay, but I'll at least take us through this example before we contemplate breaking further. So we ran the simulation, we make the plot again. Oh, Sanan. What happened?
What's up?
It broke. No, I was just teasing you. It hiccuped. It didn't display the.
Sorry,
window.
I was just responding to a lot of messages on
No,
Slack. So
I'm sorry to bug you. Yeah, no, I was teasing. Every once in a while, something goes wrong, and it doesn't display. And the easiest thing to do is just to close and open it again. I have not personally encountered an instance in VS Code where the application shut down. Rulebender used to do that all the time. It would, after a certain number of plots you'd make, you try to make the next one, and it would either freeze or shut down. Fortunately, it would restart in almost exactly the same place, so it wasn't a big deal. And VS Code will do the same thing if that happens. But okay, so now we have our two plots. I can look at them sequentially, but it's hard to see, compare them directly. So if I want to compare them directly, I can split the window, move one of them to the other window. Oh, yeah, I've done that before too. Yeah, oh, you have to, there's probably an easier way to do this even. I had closed one of them. Darn it. All right. Well, let me just fix this one while I'm here. I managed to split it multiple ways, even I don't know why. Auto scale this thing. Someday we'll make it automatically auto scale, but I don't know how to do that at the moment. Close that one, open this old one that we had before. Put it in the wrong place. And then plot this one. Oh. Oh, the best laid plans. Okay, I managed to plot the same one twice. This is the. All right, man. This is the right one. But the window splitting is acting very strong. I'm gonna just go back to like a place that I know. Get rid of this. Try splitting this again. In this one, I'm going to open the first GDAP file and plot it. And it makes a nice plot that's auto scaled to the current window size. Let me go to this one. I'm gonna open the second GDAP file and make a plot. And there we go. Okay. So now I have my side-by-side comparison. And you see that I made the terminal window go away. It'll come back if I run another simulation, or I can, oh, I forgot how to get the terminal back. There's a way. I think if I go to BNGL, no. It is getting close to lunchtime because I'm losing my mind. There's a way to make the terminal come back. I have forgotten it completely. No. Anyway, I'm not going to worry about it. This was the point here was to show side-by-side comparison. We did that. Oops. But obviously this is if we want to systematically look at how a parameter affects the behavior of a model, we need to do a parameter scan. And so I think this is, I think we'll end, I'll just show the parameter scan. And I think there's an exercise based on the parameter scan, and I'll leave it there, and then we can break for lunch and then come back rejuvenated and check out people's answers to the parameter scan, show what happens, and continue on our way. So here we just want to systematically, so we saw one R0 value that was high and one that was small, and they behave differently. Let's systematically look at what varying R0 over a range of values does. So the other thing that I wanted to show here, how do I manipulate files in the interface? So before we do the parameter scan, maybe it's a good idea to copy the model and call it something different to indicate the version that we're using for the parameter scan. So to do that, you can right click on any file in the interface and it'll bring up a series of options. Here we just want to do copy. So we select copy, and then we go back into this area here, right click and oh, what happened? I missed something. Right click copy, then right click in the area below here, and I don't know what's going on. Oh, that was really weird. That I'm getting a right click from PowerPoint. Right click in the area below, by which I mean down here. Select paste. Somehow I missed one of the screenshots here. And then when you paste, you'll get a copy. So I got the copy when I selected paste. So right click copy, then right click paste. We get the copy, we rename the copy, scan, okay? And then we'll edit the scan. We can edit the scan file. Let me just get my windows back in here. So what I'm showing here, we right click, we copy, right click again, paste, we get a copy of the file, which we can rename. You can actually just hit return when you mouse over it, and then call this scan. So now we have the scan file, and we can comment out we could just delete the simulate command, but it's also just as easy to comment it out. And now I remember that there's a thing I can do called parameter scan. So I type PA and snippets magically pulls up parameter scan, which I can select. And then that gives me a form of things to fill out. So the key thing I have to set for parameter scan is the method, the simulation method. We're usually going to want to use ODE for parameter scans, but not necessarily always. So we hit ODE. Then for each simulation, so what this is going to do is it's going to run a simulation of my model, a new simulation for each value of the input parameters, scanning over one of the input parameters. So here I'm going to scan over R0 over a range, and then run a simulation. T-start is by default 0, so I actually don't need it. So I can just actually delete this one. T-end, I have to cheat and remember what I set these values to. Here, it just needs to be set to something. Yeah, here are the values that I picked. Something large, 500, so that we're running the simulation for long enough that the outbreak has ended. That we've gotten to a steady state. And we're gonna look at the total number of recovered individuals. So the total number of people who got sick in my outbreak as a function of the initial R0 value. The number of steps is also optional. Each time course that I'm going to compute is computed over a certain number of steps, just like we did when we used our simulate command. It doesn't actually matter for the parameter scan plot that I'm going to make. All that really matters is the end time. But if I want to go and look at individual trajectories, it's nice to have them available over a series of time points. So I picked 100 here. And then the key part of the parameter scan is the actual parameter that I'm going to scan. So notice this is all on one single line of code. And I can tell that the first line ends with a comma. The highlighting continues for the entire line, and the final parentheses occur at the end of the line. VS Code is perfectly happy to have the lines be as long as they want to be. I'll show what happens in the interface. We have to toggle one of the editor parameters in order to make it look like this. Otherwise, it'll just go off the end of the screen. Okay, so and then we have to specify the parameter we want to scan and the range that we scan it over from. So from 0 to 4, and the total number of points that we want to look at here. I did 100, and just to make it reasonably smooth. And the log scale is sometimes very useful when you want to scan a parameter over many orders of magnitude. Then if we invoke log scaling, it'll be much easier to see the how the parameter variation occurs when at small values, sort over the full dynamic range. It's a very useful trick to scan parameters over a large range, to use the log scaling, but we don't need it here. So so if I go back and enter the values that we had, 100, I don't know why it stopped highlighting. And we went from 0, 4. Oh, because there's 100. We don't need log scale. We'll just clip that out. And we see here in my window, the wrap is not set. So it just keeps going. There's a very long line. It goes over the edge, goes off the screen if I have the screen set to a smaller size. So it's useful. You can toggle on word wrap. I guess it's Option Z, toggles it on and off, and then you can display the whole command on a single in the window. You can't, in BioNetGen, syntax allows you to continue a line by using a / character, but the VS Code extension doesn't handle that gracefully, so we don't use it anymore. I can't even type it. Yeah. So what happens if I try to continue a line with a /, and you'll see this in some of the files, some of the example files. This is still fine. BioNetGen will still run this model, but the syntax highlighting is broken. Turns out not easy to fix that problem at all. So we haven't tried to do that. So we have our parameter scan set up, and then we can run it as we did before. It'll hiccup just a little bit longer, and you see that a whole bunch of stuff happened. And in particular, we generate a new GDAT file for every different value of the parameter that we had set. So we have 100 of these files. And those files are all stored under the subdirectory surscan. And there's only one subdirectory that has this timestamp here. Sure you're running. Yeah, okay. And then this subdirectory contains another subdirectory that has all of the scan files that we just generated, so all the GDATs, and then for each one there's a CDAT file and also a net file for all of these, which could be useful for looking at individual trajectories later on, but for now we'll just leave it put. And you'll notice that the interface, the runner did not bring up the plot by default. I forgot why, but it doesn't recognize scan files just yet. But if we click on it, we can see that it's there. It's a tabular file just as before, and we can plot it now and see on the x-axis is R0, and on the y-axis are the final values of each of the observables SI and R for each trajectory. So at the end time, 500 time points, what is the number of susceptible, infected, or recovered individuals? So we see that the infections have essentially died out for the overall the full range of R0 by 500 time points, or 500 days, I guess, whatever the units are in this model. And what we're interested in actually is just the curve of the recovered as a function of R0. And so this tells us the fraction of the population out of 100 that were infected and have now recovered as a function of R0. And what we see is that there's basically an inflection point around R0 = 1, where below that there's low infection, low number of recovered individuals, and above that there's a high number, and it increases with R0. But interestingly, although we typically think of R0 as being the threshold below which the infections are going to die out, in a small population size, if a substantial fraction are infected, that a substantial number of people will still become infected. So the infection doesn't completely die out even at an R0 of 1 in this example, and that's what we got out of our parameter scan. Yeah, the log scale parameter scan we will come back to at a future time. Basically, the use case for a log scale parameter scan is when you have a parameter that varies over many orders of magnitude. So if you have a parameter that varies, say, between 0.1 and 1,000, and you make a plot of you vary the parameter on a linear scale over that range, you'll see a lot more parameter values that are close to 1,000 than are close to 0.1, and so you won't really be able to tell what's going on for small values of the parameter range. So it's a way of sort of zooming in on what's happening at smaller values of that range. It's very useful for rate constants, because rate constants, we typically, when we want to look at how the behavior of a system changes with the rate constant or an affinity, then it can vary over several orders of magnitude, and it's much easier to see that in log space than in linear space. Okay, so here I point out that, yeah, substantial fraction was infected below R0. So the exercise is to increase the number of individuals in the population to 10,000, and to do the parameter scan again and see what happens. So I think I will stop there. It's very hard on the fly to remember if you're keeping on time, because my clock says it's 1:54 p.m.
Yeah, I guess
we're over, right? Over the schedule.
Well, a little bit over the schedule. How long has remained from your presentation? Is it finished? Because I guess we have a lunch break scheduled at 12 to 1. So
that's it. Yeah. Well, that's at 3 p.m.
Yeah. So, and we had one break which we didn't go to. So if you want, we can just like come back to the new Zoom in like 20 minutes. Is it enough? Or, I mean, what do you suggest? It depends on how much you have in your presentation. How much time do you need for the next presentation? Like
I'm
1
thinking,
hour?
I'm trying to think of, so I know that I apologize for not being terribly organized on the time front. This is the first time I gave this one for VS Code. So the timing, I'm not good at the timing. And what I'm trying to think of is, I know some workshop participants schedule their involvement for particular times. So I'm just trying to think of getting back on schedule for BioNetGen at 4 p.m. Eastern time.
Yeah.
So what I would suggest is let's come back at 2:30 my time or 12:30 PDT.
Okay, cool.
And that, and then we'll go through the rest of this part of the.
Are you calculating correctly? You mean right now it's
It's almost 11 in
It's
the
a-
Pacific.
yeah.
Pacific
Did I get it
Coast.
wrong by an hour? 11. I meant
Ah.
11:30. I meant 11:30. Yes.
So
Okay.
come back in 30 minutes, you mean, yeah?
Yes. Yes. I meant 11:30. My apologies.
Okay, okay. So then if you like, we can go to the new Zoom if it is okay for you, because if the each Zoom link, you know, each Zoom just is very long, then we had a little bit of problem for cutting and putting in the YouTube for the recording, I mean. So if it is okay for everyone, we can come back to the new Zoom link. I will share it in the Slack, in the general channel, but you also can go and find the second Zoom link and click on this in half an hour. So, okay, everyone agrees?
Yeah, we'll do that. And what I just to finish, I will start talking instead of coming right back to the VS Code stuff. I'll give the presentation on rule-based modeling first, and then we'll come back to interactive as time permits. But I'm going to try to get us to 1 p.m. for BioNetGen, and let Sinan take over for there, and then we'll have some more time in the afternoon to finish up on the other aspects of different modeling features in VS Code. Does that sound good?
Perfect.
Yeah. Okay. Great.
Okay.
Thanks, everyone.
Thank you, and see you in half an hour.